So we had chapter 14, it's Mendel and the gene idea. So this is part 2. And the first thing we're going to talk about is degrees of dominance. So in the last video, Sophia talked about different like heterozygous organisms and how those traits get passed down. And what I'm going to talk about in degree of dominance is that the dominant allele does not always come through as you would think it would. So for a little bit of review, can someone tell me what heterozygous is? Tony?
Two different alleles of the same section of trait.
Okay. And then can someone tell me what a genotype is? Maggie, do you have it?
Isn't that like the genetic makeup, like the letters?
Yeah. And then the phenotype. Maggie?
The physical trait?
Yeah. So I wanted to review that because those 3 things are the kind of most important thing to think about when you're talking about degrees of dominance. So as we go in, I'm going to say that there are 3 different types of dominance. There's complete dominance, incomplete dominance, and codominance. And each of these concerns the phenotype of the heterozygous alleles. So complete dominance is kind of the classic form of dominance that we generally think of when we think of a heterozygous organism. With the heterozygous genotype, we get a dominant allele that is expressed in the phenotype, and only that allele is expressed. So, for example, if we had someone who is heterozygous for brown eyes, so they have a dominant allele for brown eyes and a recessive gene for blue eyes. When they mix, they would end up with brown eyes as their dominant, and that would be true in the heterozygous form. So even though there is a blue eye allele, it is being suppressed by the brown eye allele. The next form is incomplete dominance. And incomplete dominance is different because neither allele is dominant, and therefore neither one is fully expressed. So this means that the heterozygous genotype results in a mix of the 2 traits in the phenotype. So this often can create an entirely new phenotype. So for example, if we have a homozygous red flower and a homozygous white flower, neither one of these traits are completely dominant, and that's why it's called incomplete dominance. So when they mix into the heterozygous form, you get a mixing of the 2 phenotypes, and that results in a new phenotype of a pink flower. The last form is codominance, and this is when both alleles are expressed in the phenotype, so both of them are equally dominant. And both alleles share the dominance, which then results in both traits being seen in the phenotype. This is different from incomplete dominance because in incomplete dominance, the phenotype mixes the 2 traits as opposed to codominance where both one is expressed sort of individually. So if we have a white bear and a black bear, they would mix and form a panda bear. So they're not mixing the colors, so you don't get a gray bear, but you get a bear with black splotches and then white splotches. So each one is expressed on its own. Yeah?
Given 2 pairs of homozygous alleles, and you mix them, how do you know if they're going to be codominant or incomplete dominant? If they're both, recessive or dominant?
It depends on the type of, what the genes were to begin with. So, in this case, if you had the codominance, when you mix 2 of these heterozygous pairs, it would depend on how they end up. If you get 2 homozygous black genes, then the bear would be just black. If it mixes and it's heterozygous again, you get a bear that has both colors again.
It varies from example to example too. Different genes from different animals act differently. So with the flowered example, that's a specific type of flower in which incomplete dominance occurs, and then codominance occurs in different type of alleles, and sometimes it's not even the example, it's the type of trait. So a specific trait in that animal will act that way. That's just the way that the alleles work together. Because sometimes it's not even that, in the Tay-Sachs disease, it's not even that a homozygote, it's not even that a dominant trait is completely suppressing the recessive trait. It's just that there's enough enzymes of the dominant trait being made so that it like overrides the amount of defective enzymes being created. So it's just
Yeah, did that answer your question, Jeff?
Yeah.
Okay.
I was gonna just add, blood typing.
Yeah.
If you receive an A allele from one parent and a B allele from the other parent, then you're equally type A and B.
Yeah.
Neither one is masking the expression of the other, so they're both equally and fully expressed, and you get A proteins and B proteins, so you're therefore type AB blood.
Yeah, and that's the most common example you'll see of codominance is you'll see it in blood type. I mean, animals sometimes get it and you can see different splotches of color, but for the most part, it's usually blood.
So now we're going to talk about law of segregation, which is one of Mendel's two really important laws about heredity that his pea plants helped him deduce. So his observations of expressions of traits over the course of his F1 and F2 pea plant generations provided for the deduction of two fundamental hereditary principles. First one is law of segregation, which states that two alleles for a heritable character segregate during gamete formation and end up in different gametes. So let's see this visually. So Mendel's law talks about how during meiosis, after crossing over, which is when the homologous chromosomes cross over, so that there's a varied type of allele being inherited, they segregate independently of one another into the opposite sides of the cell, so that there's like an equal chance of inheriting like a YY and a little y in the next generation of organism. Who has questions about that? Okay, so does anybody know? Hold on, I gotta hide it. Otherwise, everybody will just look at the picture. Okay, so does anybody know where this occurs? Is it in mitosis or meiosis?
Meiosis.
Yeah, meiosis. Okay, let's move on. So the previous model of heredity suggested that offspring were a blend of two parental genomes, such as incomplete dominance would show, whereas if you blend a white flower and a red flower, you get a pink flower. And although that's true, it's not like the fundamental form of heredity. So Mendel's experiments obviously proved that this theory was incorrect. That was supposed to be a picture, which we just went over. So each plant in the F1 generation had a flower as vibrant as in the P generation, which showed that it's not always the blending that matters. If you mix a white and a pink flower, the purple plant and the white flowered plant of pea plants, the purple is going to be just as vibrant, and there's also going to be some white if they're not truly breeding. In the blending theory, the colors violet would have mixed together to produce a pale purple flower. That's not true. And in the F2 generation, the trait of a white flower petals reappeared. So if you mix two heterozygous purple flower plants, a white flower happened, and at about a fourth of the time, which helped him deduce this principle. So Mendel assumed from this outcome that the trait had been masked by what he termed a dominant trait in the F1 generation, and the white color was then a recessive trait that was hidden when a dominant trait was present. The F1 generation were hybrids that contained both a recessive and a dominant allele, allowing for this F2 generation to have both white and purple flower plants, despite what they mated with. So Mendel utilized substantial sizes to come up with the accurate figure for this event, which is the ratio of purple, which is dominant, to white flowers. In the case of a hybridization cross, was cross-pollination was 3-to-1, or 1/4 chance that there was going to be a recessive trait expressed. So as stated, Mendel concluded that there were alternate versions of genes that accounted for the variation in inherited characters, which were termed alleles. and this is actually pretty interesting and cool that he came up with this because they hadn't even discovered that chromosomes existed at the time. So the fact that he thought about this was pretty outstanding. So the two alleles coexist in DNA, as we've seen, and one is on each homologous chromosome and in their respective locus. Locus is the positioning of the allele on a chromosome. So the variation in nucleotide sequences accounts for the difference in alleles. So there's not like a little t and a big T on a chromosome. That's just how we express it. It's so that it makes sense for us. The little t actually symbolizes a sequence of nucleotides that codes for a dominant phenotype. For each character, an organism inherits 2 copies of the gene, i.e. 2 alleles, on a homologous chromosome, each which has 2 pairs of chromosomes, and 1 coming from each parent. The word homozygous, if you guys haven't picked up on that already, is when an organism has a pair of identical alleles for the particular character trait, which can be either recessive or dominant. You can have either big T's or little t's, as long as there's the same, they're homologous. Heterozygous is when an organism has differing alleles for a particular character, and heterozygous produce an organism with differing alleles, unlike bred homozygous. Monohybrid cross is when an experiment in which only one trait is being investigated. A dihybrid cross occurs when 2 traits are being investigated. So Mendel didn't only just study like what happens when you mix 2 round pea plants. He also studied what happens when you mix a white flower with round peas and a purple flower with wrinkled peas. And this helped him come up with the law of segregation, that they segregate independent of one another in meiosis and thus are inherited independently. So it's, you can't, have we gone over Punnett squares yet?
No.
No, okay. Well, we can talk about that later. Okay. Who's independent? Okay.
So the law of independent assortment, it's another one of Mendel's laws that he created. And so essentially what it is, so when genes are like really close to each other on the chromosome, we like associate them together. And so what happens in that association, when Mendel was saying that it doesn't matter how close they are to each other, that they're gonna sort out in meiosis independently. Like it's not, one isn't gonna affect the other. And so what a dihybrid is, it's an organism that's heterozygous in both aspects, and I'll show this later. But so like it says, so basically what it is, that it's in gamete formation that the alleles themselves are going to separate out in random order. And so the genes are gonna, so like these alleles, you can have any allele combination as long as like each gamete is gonna have 1 gene for each allele. And so, but the truth is, I guess, like we've gone further in science, we found that this law isn't as true as we used to think it was, because when genes are close to each other, it does affect how it affects gamete formation and how genes are assorted. And so if we kind of take a look at this example, back again to the pea plants. Okay, so again, the pea generation are 2 pure plants, 2 dominant, so this would be homozygous dominant, homozygous recessive. And so this F1 generation here, this is going to be considered your dihybrid because it is heterozygous in respect to both genes. And so what the law of independent assortment really talks about, what it's saying is that you're not only going to get, you can't only just be homozygous in both aspects or heterozygous in both aspects, but they can mix and you can make 4 different combinations out of here. And so then this would be your F2 generation when the 2 plants are like pollinated. Okay, so is the law of independent assortment, is it always accurate? Sean, what do you think? Is it always accurate or no?
Relatively.
Relatively, okay. As long, what is like the requirement, like what's necessary for this, like, what does each gamete have to have in order for this law to make sense? Like each gamete has to have, Sam?
2 genotypes?
Well, no, it has just has the one genotype, it has to have one allele to make sure, it's, when it's independently assorted, each one's gonna have at least one allele.
Alright, so for pedigree analysis, geneticists aren't able to manipulate the mating patterns of human. So does anyone know what they would have to do? What? Oh, yeah, I know, but I don't want them to see what I'm gonna say. So does anyone know what they would have to do in order to like be able to guess the outcomes of patterns that would show in like offspring from two people? Sam?
Trace like ancestry.
Yeah, so they'd have to analyze the results of the mating that happened with like, say, a mother and a father, and then trace it through like maybe 3 generations to see what traits are shown in those offspring. And in order to do this, like Sam said, they have to collect a family's history for a particular trait. And assembling this information into like from a family tree and describing the traits that are shown in the parents and the children is done in the form of a pedigree. And so in this picture, if we look over here at the keys, the squares are male and the circles are female. And the ones that are colored in this case purple, would be the affected male and the affected female, and the ones that are left blank are just, they're not affected. And then a square and a circle means mating, and then a circle and then like this means the offspring that happened. So in this particular picture, it shows 3 generations of this pedigree. It shows the, say the grandparents or whatever, and then the 2 parents for these 2 here, and then the offspring. And we're tracing a widow's peak in this case. And the dominant allele would be the capital W, which would mean like the dominant allele would be to have a widow's peak. And then the recessive allele would be the lowercase w, and they wouldn't have a widow's peak. And like Sophia and Kate said before, you can have either a double capital W for the dominant allele to show through, or a homozygous, heterozygous here for it to show through, and then the recessive would be just 2 lowercase w's, yes.
What do you call the 2 capital W's, what genotype, how would you label that genotype?
How would I label the genotype?
Yeah.
Like, I would call it a homozygous allele for, homozygous dominant for widow's peak, sorry.
And
Okay.
then what about the 2 small case W's then?
That would be your homozygous recessive. Okay. So, in this case, the 2 grandparents, this person's parents here had heterozygous for a dominant allele, heterozygous dominant for the widow's peak, and then homozygous recessive for the widow's peak, and then the same case here. And then it showed the offspring for a heterozygous,
Stop the